Good morning, everybody. Yesterday, I presented a video on KCSE Biology Practical for 2012, but I saw there was need for also producing a video on the confidential requirements for that paper, because I thought if that video on the paper does not go hand in hand with the confidential requirements, there might be some misunderstanding of the requirements, and therefore, if you are going to watch that video, please have a look at the confidentials first so that you know what was required for that practical. Welcome, guys. So, the requirements for question 1 were there was a photograph in the question paper, and the photograph was of dicotyledonous seedling during germination with bending hypocotyl as shown in this diagram. Question 2, you are supposed to give the students a bread mold which was labeled specimen D or Rhizopus species. Then you are also supposed to be having a mounting pin, white tile, and hand lens. To prepare the mold or capturing the Rhizopus or preparation of specimen D, the Rhizopus can be captured as follows. Step 1. Moisten slices of bread or any other appropriate substrate. 2. Add a little sugar, sucrose, to the slices to hasten growth of Rhizopus. 3. Expose the slices to air for a day. 4. Cover the slices to exclude dust. And 5. Place the catcher in a warm place and leave it for 5 days until the Rhizopus is fully grown. So by the end of the 5th day, the Rhizopus will have grown if you follow those 5 steps. Note, the catching should be properly timed to ensure that the Rhizopus is fully grown on the day of the examination or on the day of the practical, as the examination council timetable will indicate. So you time backwards from that day, all right, so that the Rhizopus will be fully grown before you give the candidates. In B of question 2, there was a photograph of bougainvillea flower showing different parts of a flower. And the photograph, one of the flowers was labeled E. There is a label E there. Question 3, each candidate required or requires for that examination 110 mils measuring cylinder, white tile, grass rod, stop clock or watch, 2 cubic centimeters glycerin labeled washing up solution to prevent spilling over of bamboos from foam, 1 fresh Irish potato, as couple 5 cubic centimeters of dilute hydrogen peroxide solution, substance labeled F, which is a weak acid solution of pH 4, substance labeled G, distilled water of pH 7, substance labeled H, alkaline solution of pH 9, access to distilled water in a ratio of 3 candidates to 1 wash bottle. And finally, you need a dropper. Preparation of the dilute hydrogen peroxide was you mix 20 volume hydrogen peroxide with the distilled water in the ratio 1:1. So that is for the teacher. So especially when you look at the video on the practical, the way you tackle the questions, you'll be wondering now what is washing up solution. Here you have seen it is 2 cubic centimeters of glycerin, where you are adding to the setups. There were 3 setups according to the substances F, G, and H. So after watching this video, you are more informed. So that is the end of that video. Please don't forget to subscribe to my YouTube channel for more interesting videos.